The gap genes, along with the maternal effect genes, activate the expression of the pair rule genes, and these are also transcription factors. These genes begin to be expressed at around the 13th cell division cycle, so this is when cell membranes are beginning to form between nuclei. And segmentation first becomes apparent as these genes begin to be expressed. The expression of the pair rule genes is activated, as I mentioned, by the gap genes, but also by the maternal effect genes. As you can see from these pictures, the pair rule genes are expressed in 7 stripes along the anterior-posterior axis of the embryo. And they're expressed in such a way that one row of cells expresses a pair rule gene and the next one does not. So each pair rule gene divides the embryo into 15 domains if we count the regions where that particular pair rule gene is not expressed. For example, this staining shows the expression of 2 different pair rule genes, even skip, which is shown in this orangey red, and FTC, which is shown in the black. As you can see, each of these genes is expressed in 7 stripes, but these are not the same stripes. Different pair rule genes are expressed in different stripes along the anterior-posterior axis of the embryo. This means that the combination of pair rule genes expressed in a given stripe will give the cells in that pair segment a particular identity. The expression of each stripe of a pair rule gene is determined by different regulatory regions, which your textbook refers to as enhancers. Each regulatory region contains binding sites for transcription factors, and some of these, as I mentioned, are going to act as repressors and some are going to act as activators. Usually, one enhancer drives expression to either 1 or 2 stripes. So for example, in this figure, the different enhancers are color-coded. Some of these regulatory sequences are located upstream of the coding region, which is right here, and others are located downstream of that coding region. The enhancer shown in yellow drives expression to the first stripe, and the one shown in blue drives expression to the 5th stripe. So the bottom set of pictures here shows even skipped protein stained in orange with antibody staining, and these embryos were also injected with transgenes containing the enhancers for either stripe 1, stripe 5, or both stripes fused to a LAC-C reporter gene. The pattern of LAC-C expression, which is shown here in the dark blue, shows how each enhancer is driving expression to a different stripe in the embryo. The last picture on this slide illustrates the relationship between the pair rule genes and the gap genes. Recall that the expression of the pair rule genes is activated by the gap and the maternal effect genes. The embryo shown in this figure is a giant deficient embryo. And remember that giant is expressed in 2 broad stripes, one near the anterior of the embryo and the other one at the posterior. In the giant deficient embryo, the expression of even skipped in stripe 5 expands. So what does this suggest about the role of giant on the expression of even skipped at this particular location? The fact that the expression of the even skipped stripe expands in the absence of giant suggests that giant acts as a repressor of even skipped in this particular region of the embryo, so that when giant is not there, we see more even skipped expression. So the expression of giant determines the anterior boundary of even skipped expression in this particular stripe. Each of the regulatory sequences that regulate the expression of the pair rule genes in the different stripes have binding sites for gap and for maternal effect genes. This is an example showing the regulatory region for the 2nd stripe of the even skip gene. The activators are shown above the line here and the repressors below. So you can see the activators include bicoid and hunchback, while the repressors, which are shown at the bottom, include giant and cripple. Depending on the enhancer, different gap genes will either act as activators or as repressors to a particular pair rule gene in a given region of the embryo. So that means that the same gap gene could function as an activator for one stripe and as a repressor for another. For the 2nd stripe of the pair rule gene even skip, which is located in the 3rd parasegment, because remember that these skip a parasegment, bicoid and hunchback act as activators, as I mentioned, and cripple and giant act as repressors. If we compare the location of these genes in the embryo, we see that even skipped is expressed where bicoid and hunchback are present, but where giant and crupple are absent. If crupple were deleted, the posterior border of expression of even skip would be displaced or expanded further back. So to summarize, what controls the expression of the pair rule genes are the cis-acting elements, which are the regulatory regions in the DNA, and the trans-acting elements, which are the transcription factors, the products of the gap genes and the maternal effect genes. But there are some pair rule genes that are not regulated by gap and maternal effect genes, but instead they depend on the expression of other pair rule genes. Of the 8 pair rule genes that have been described, 3 are categorized as primary, and these are regulated by the gap and the maternal effect genes, and the rest are categorized as secondary, and these are regulated by the primary pair rule genes. Once the pair rule gene expression has been established, these genes will then go on to activate the segment polarity genes.